UK in the last 2 weeks, it has been the Wimbledon tennis tournament, and traditionally it rains throughout Wimbledon, and this year has been traditional in this respect, so it's very nice to be invited here. Thank you, Miklos, to Szeged, where it is beautifully warm and sunny. The city is very beautiful too, so I'm delighted to have a break from the rain in Cambridge at the moment. I'm delighted to be invited here to talk about some super resolution microscopy techniques. I am an applied mathematician. I study the mathematical and computational techniques which are used to do the super resolution imaging as Miklos has described. And what I'm going to do in this talk is I'm going to talk briefly about what is it that we want from super resolution microscopy. What do we want our mathematical techniques to do? Because this is what really interests me the most. And then I will talk about some of my research where we've used some super resolution microscopy to study antibiotic-resistant bacteria. I will talk about 2 or 3 different super resolution techniques. One of them will be STED. This is what I call a direct super resolution technique. Basically, in STED, if you set out to do STED, you're saying, well, I'm willing to spend a lot of money to build a better optical system which will take better image data directly. And that has some advantages. You can apply it to many different things. And then I will talk about mathematical methods for doing super resolution imaging. If, like me, you can't afford or you don't have steady enough hand to build a very advanced STED microscope, then instead you can take conventional image data, and sometimes it's possible to use mathematical techniques to take blurred image data and reconstruct what the better resolved image should look like, purely using inexpensive hardware and maths. So we'll talk about this ELM technique, which I developed. This is ellipsoid localization microscopy. It's similar to the single molecule localization microscopy that Miklos described, but works for things like these ellipsoidal fluorescent shells, different objects. And I'll talk briefly at the end about structured illumination microscopy, which is a Fourier technique. And I won't go into the details of Fourier transforms because I've never really liked Fourier transforms that much. I could work in real space. I'm an applied mathematician. So Fourier transforms are a bit harder. Now, just zooming out briefly, why is super resolution, why do we want to do visible light super resolution microscopy? Well, this is because optical microscopy is absolutely essential. It has been and it still is. It's an essential technique for all kinds of biological and medical research, for the main reason that light can travel through the air and it can travel through water, which means it can basically travel through living tissue, at least to some extent. So we can use light microscopy to look at, so we can look inside biological samples, and we can look inside samples when they're living. And on top of that, that's so useful on its own that there are many standard techniques for fluorescently labeling particular chemicals or proteins inside living tissue in different colors. So this just shows a cell. This is a very standard sort of image. It simply shows in grey a transmitted light image of a cell. In green and blue it shows 2 different virus proteins which we were interested in studying, and the red colour is just a nuclear membrane stain. And this is related to a question, this is for a particular virus, herpes simplex virus. The question is, where is this virus manufactured in the cell? Well, we think it turns out that some of the virus, the proteins that make up the virus, are manufactured inside the So if we want to look at topics of modern interest, such as these are the amyloid protein fibrils, which Miklos showed you. Well, they're not the same ones that Miklos showed you. So these structures are about 1 micron in length, and the formation of these structures is associated with the development of Alzheimer's disease and with other neurodegenerative diseases. So we want to know if these structures have been formed, and with super resolution microscopy we can do that measurement inside cells which are, well, maybe living. In any case, we can do that measurement inside cells. Previously, if we needed to use electron microscopy or atomic force microscopy, those would be techniques that we have to do under vacuum. So you can't look at biologically important chemicals in the native state. You certainly can't look at them in living systems. You can't even really look at them inside cells. You have to do some sort of invasive preparation and purification of the sample, and you never know whether that's completely changed the state of the protein that you have. So we want to get away from doing high resolution techniques under vacuum, and we want to obtain the fact that in fluorescence microscopy we can do chemically specific labeling, whereas in electron microscopy we have very little ability to tag different proteins and say which is which, but that's something we can easily do in light microscopy. Okay. So in principle we're going to take images of things. This is actually a bacteria spore related to some antibiotic resistance research that I'm interested in. But part of my research is, well, we take images. Actually, an important part of my work is what I call quantitative imaging. So what we want to distinguish an image from a conclusion that we need to write in some scientific papers. So there's a saying in statistics that data is not information, and information is not knowledge. And we need to, in order to do publishable biological research, we often need to obtain some new knowledge about a particular system. And if we take some pictures, what I want to do is I want to take this image data and I want to learn some information from it. One way we can do this is just with a visual assessment. I can visually look and say, aha, this spore is, well, it's spherical. I suppose I could publish that. But it would be much better if we have not a visual assessment, but a mathematical assessment for taking the image data and doing some consistent calculations on the image data to get some more precise information. And in particular, the things I'm interested in doing, I'll come back to this graph later in the talk. I'm interested in mathematical techniques which, because they're computational, you know they're consistent. You're applying exactly the same analysis to every image measurement. They can be accurate because the computer has a lot more time on its hands to painstakingly look through thousands of different images, which you could never do visually. And another thing that I want my image analysis techniques to be is that they should be useful. So I should learn some information like the radius of a particular protein layer in the spore. And then hopefully that will be useful for me to write up some research on that. And the key mathematical technique that I want to use in order to do quantitative imaging is called inference. And in mathematical inference, what we say is, well, we have some data, and the data has a certain amount of information about the sample in it, but we're going to add some more information. We're going to assume that the sample, it obeys some sort of equation. So I'm going to assume that the sample is, say, a spherical shell or some other shape. And given that we know we've added some extra assumptions about what the sample is, then we are able to get an equation which describes what the image should look like. And often by fitting the equation to the image data that you actually get, you can obtain much more precise information on something like the radius of the spore than we could obtain if we just looked visually at the image data. So that's the point of what I want to do. I want to do mathematical inference. I want basically an excuse to do some equations and then to fit the equations to some image data and do this on a large scale in the computer. Okay. So that's the principle. In principle, I'd like to do very accurate, useful measurements in super resolution microscopy. Before we do anything useful, let me give you something which is not useful, but I do think it's interesting. As an example, I showed to my undergraduates. So this is given the following image, which is actually a painting I like, and it was painted in 1835 in the Industrial Revolution. Can you tell me, given this painting and some extra information, what time it would be if you were to look at your watch when you see this painting? And here this is a painting by William Turner, English painter. And so he was very famous for being very good at painting light and water. And this shows a good, so I'm from the Department of Chemical Engineering, this shows a good chemical engineering scene because it shows some people transporting coal. And they're taking coal from one ship which has come down the river, and they're loading it onto ocean ships so they can transport the coal to sell it in London. It's a good chemical engineering picture, I tell you. And so this painting, this is painted in Newcastle. So to give you some extra information, this scene here is painted in the industrial city of Newcastle, which is on the east coast of England, and we're looking in this scene eastwards into the North Sea. Okay, so that's where it is, and that's what the painting looks like. So, can you tell me? I tried to get my undergraduates to volunteer answers to this question. If I had a watch, the watch was invented in 1835. They weren't very good, but if I had a watch and I looked at it, what could you tell me about the time from the fact that you're seeing this scene? Would anybody like to volunteer what time it might be from the picture?
I don't understand the question.
Aha! So,
If time is already there.
so the year, I've given you the year. Yeah, I want more than the year. So if I were to look at my watch, and if I wanted more precise than the year, then how would you go about obtaining that information? So that's an example of inference, that we have some image data, and possibly by adding some other sorts of equations that we might know about, or some other kind of information that we know about, we want to reach a conclusion as to what time it is. Does that make sense as a question?
Surely not sunset.
That's right. So that's good. Thank you for that answer. So you're thinking, because we're looking east, you'd be looking at. So I tried to lure my undergraduates into this answer. So I tried to get my undergraduates to say, we're looking east. So okay, this must be a scene of the morning sun rising over the sea, and therefore, given the altitude of the sun above the horizon, maybe it's about 9 AM, maybe. Okay. And so I tried to get the undergraduates to say that because it's wrong, and give them something to think about. So this is an old painting, and the colour has faded in the painting. So you can look at this and you can say this is a picture of the sunrise. So actually, the full title of the painting is that this is people transporting coal from river boats onto ocean ships at night. And you can get this purely from the image data, because so in the image you've got these people are working by torchlight, and even though the colours faded quite badly, you can see that the people are casting shadows over the water. And in order to cast dark shadows, it can't be during the day, not if you expect to see shadows from torchlight. So you see, this must be at night, and therefore this is actually a scene by moonlight. I think it's a quite nice scene. And this is, I think, a good example of quantitative imaging, because I can be much more accurate than you might think. So this scene, it's ambiguous as to whether this is the sun or the moon, because it's a full moon. And I've told you the year is 1835, so there are 12 days when there's a full moon in 1835. Okay. And we know also, we know that the moon subtends an angular diameter of about ½ degree when it's viewed from the Earth. But we also know that that's not how people perceive the moon. We know that people perceive the moon to be about twice as big as it really is. So people think the moon is about 1 degree. So we'll assume it's been painted at 1 degree, because the painting should be what you perceive. So the altitude of the moon above the horizon is about 10 degrees. So if it was the sun, at this point, we could plug in 10 degrees altitude into an equation, and I can tell you exactly what time it is. The moon is way more complicated, because it's simply not locked to the solar day properly. However, what we can do is we can get some astronomy software from the internet, open source, and we can simply plug in the year 1835, and we can plug in the location, so Newcastle, and we can scan forwards until the altitude of the moon above the horizon is 10 degrees, and we can do that for the 12 possible days when there's a full moon, and it comes out, so my preferred date for this is February 13th, 1835, 6:30, and it has to be February or December of that year, because any other time it would actually be daylight when the moon is at that height. So we can be quite precise from the image data. We can learn some quite precise information about what is going on. We can say that it's 6:30, and depending on which day it is, it's 6:30 plus or minus about 15 minutes, which I think is a nice example. It's not very useful for writing scientific papers, but it's a nice example that sometimes you can learn more information from a picture than you might expect. Okay, what I actually want to do is talk about super resolution microscopy. Are we having coffee at 10:30, is that right? Yep, okay, good. Miklos and I, about 8 years ago, 9 years ago maybe, 8 years, it might be 7, 7, started building up these super resolution microscopes in the group of my boss, Professor Kaminsky in Cambridge. And Miklos actually has a good hand for optics, and I did some work on some of the software for our techniques. And so more recently, a friend, a colleague of mine who was a microbiologist asked, can we use one of your boss's expensive super resolution microscopes to measure the shape of some bacteria that he's interested in for the purpose of studying antibiotic resistance. And I said, yes, we can. We can use an expensive super resolution microscope to look at your samples. But it would be much better if we could use a cheap, ordinary microscope of the type that everyone has, and a super resolution mathematical method that I've been working on for precisely this sort of sample. And I will come and talk about that a bit later. But before I do that, I need to talk about bacteria spores, and then I will talk about the direct super resolution. So I will talk about the expensive way, the expensive route for measuring these things. So this is an electron micrograph of a bacteria spore. Bacteria spores are very interesting. Well, no, bacteria spores are quite interesting, but they're very important. So that many bacteria, this is effectively a bacteria seed. Are you a medical student? You know more about bacteria than I do. So this is effectively a bacteria seed. So that many bacteria, when they sit growing, and then they run out of food, and they can turn into these seed-like spores. And they can, the bacteria seeds can survive for a long time if they're left, so from Egyptian tombs we know that spores of bacteria can survive for 3,000 years, and then if you put them on a petri dish with some food, then within about 10 minutes they will germinate and turn into perfectly normal bacteria. So these things they're interesting just because they're very tough, and not only can they survive for a long time without food, if you put them in boiling water, you do that to disinfect things. If you put spores in boiling water, they just think they're having a warm bath, and you take them out of the water and put them on some food, and they think, great, and they turn into vegetative bacteria. And some spores, this one is a harmless spore. most of them are harmless. This one just grows in the soil. Doesn't do anything. Doesn't do anything dangerous. You can eat this one. But other spores are important in medicine. So, for example, anthrax is a spore disease. Anthrax is dangerous is because it can survive in the soil for a long time, and then it can come out and infect people. And another spore disease, which is probably medically more important because it's more common, is Clostridium difficile, or C. diff. It's typically lots of people have C. difficile growing non-dangerously inside their stomachs, but it's a dangerous disease when people are given. It can be a dangerous infection when people are given antibiotics, because the antibiotics kill all the bacteria in the system except for the spores, which are dormant, and they're protected. They're protected from chemicals by this armoured coat, and also by the fact that they are metabolically inactive. But the interesting thing from a super resolution microscopy point of view about the spore is this grey armoured coat, which protects them from the environment. Because of this and the fact they're just dormant, they survive antibiotic treatment, and then suddenly they're the only bacteria that's alive, and all of their competition has been killed. And frequently, after people are treated with antibiotics, they will acquire a C. diff infection. And because of its spore-forming nature, it's very difficult to get rid of. You can simply repeatedly give people antibiotics, and every time some more spores will have survived. So we'd like to understand more about the science of how spores are protected from their environment and how they germinate afterwards. Partly because it's important in medical research, and partly because it's just a very interesting structure. So this coat is actually made of about 70 different proteins, which are arranged in skins. So like the skins of an onion, you have some proteins on the very outside, which are sometimes they're to do with making the spore stick to some surface so that it can't be cleaned off. And then you have some proteins which prevent the spore from being dissolved by digestive enzymes. And then you have some interior layers of this coat, which are simply there to provide structure and to hold the thing together. And we'd like to know which protein does what, but then we have a problem, which is that in order to know which protein does what, we would like to have a map which says exactly where each protein is. But the normal way, as we've heard about earlier, the normal way for labeling a specific protein is to use something like a GFP fusion to highlight just one protein out of about 70. But if you do that on a bacteria spore, then no luck, because the coat is only about 100 nanometers thick, and conventional imaging doesn't have the resolution to map out which protein is where in the coat. So super resolution microscopy might be a way to do it. And it's for that reason that my colleague asked me, can we use an expensive super resolution microscope to measure this spore coat structure? So the coat is the interesting part from my point of view. But then these things are also medically important. Okay. Just one extra comment. Let me just note, these spores are roughly spherical. This one is spherical, and other ones are close to being spherical. This is going to be very useful later when we do some maths. Okay. And then I have a few slides about super resolution and why is there diffraction limitation. I will just go through them very quickly, because as Miklos says, the fundamental problem that we're looking at is the diffraction limit. That is, no conventional optical imaging system can allow us to resolve features which are smaller than about the wavelength of light. So features smaller than about 300 nanometers, we cannot resolve. This is because of diffraction-limited blurring. And there are two other possible problems as well, which might limit the resolution that we get from an experiment. But these are both sort of trivial. One problem is pixelation. It might be that we have a camera with a really big pixel size, and even if the optical image, which is formed by the optics, if it has good resolution, if we have two big pixels like this, then then we might be throwing away some information just by having too large pixels. That's a trivial problem because we can always add more linear magnification, in which case the blur would still correspond to the same uncertainty of 300 nanometers on the sample, but we could make sure that we're not limited by the pixel size. So pixelation shouldn't be a problem, but you just have to know it's there. And there's also the Nyquist limit. The Nyquist limit basically says you can't have more resolution than is actually present in the specimen. Again, that's a trivial problem because I know I'm looking at bacteria spores where I know I have lots of things separated by 10 nanometers. So the Nyquist limit says I need to have, if I want to observe some feature with a length scale of 10 nanometers, then I need to know that there's a fluorescent molecule present every 5 nanometers or more frequently. And that's because in order to see a structure with some sort of 10 nanometer scale, there needs to be a missing fluorophore that you need to be able to see it's missing. So you need to have, in theory, you need to have fluorescent labeling which is twice as dense as the resolution that you want. That's not going to be a problem for the experiments I'm going to show you, but you need to know this. This basically says you can't resolve structure that's finer than is actually there in the sample. And pixelation says don't use too big a pixel size. And the fundamental problem is diffraction limitation, that the blur is going to correspond to about the wavelength of light on the sample. Okay. So how do we beat this? So this is a slide showing how we failed to beat diffraction limitation. This is my attempt at illustrating a confocal microscope. So a confocal microscope, you take your laser beam and you focus it to the smallest diffraction limited spot that you can on the sample, and then it's a scanning method. So I have some sample which has some fluorescent molecules on it, and I scan my laser beam across the sample, and when it hits a fluorescent molecule, I get some light, and then I scan it to the side, and there's no more light, and I keep scanning it. And when there are two fluorescent molecules in the spot at the same time, they're basically too close together for the system to detect two separate fluorophores, basically. So the resolution, the diffraction limited resolution in a confocal microscope, it comes from how small can you make this laser spot on the sample. And it's a bit more complicated than that, but basically you want to make as small a laser spot on your sample as possible for lighting up the fluorescent molecules. And in this case, we've failed to distinguish these two different fluorescent molecules. So what I will look at before coffee is the expensive hardware method for doing super resolution microscopy with a better machine is a way of getting around this diffraction limit. So here are some fluorescent molecules, and imagine that I have a green laser spot, and this spot is as small as I can make it with optics. Right. So at the moment, I basically cannot resolve the difference between the difference in position between these different molecules because they're all illuminated at once. And basically, if I move the spot to the side, basically none of them are illuminated. So I'm just getting a bright blob of detected signal. So diffraction limitation says I can't make my Gaussian beam any smaller than that. But what it doesn't stop you from doing is making a different shaped beam. And what we do is we make this red donut-shaped laser beam. So diffraction limitation does not stop you from making this red beam. And so you illuminate your sample with the green diffraction-limited beam, and then you arrange the optics of the system to produce what I put here as a red beam. And the effect of the red beam is that it drives what's called depletion, so that your fluorescence molecule is intended to detect the fluorescence which comes when you illuminate your sample with green light. And a property of fluorescent molecules is that they have a complicated energy level diagram. But basically, the result of this energy level diagram is that if I illuminate my supposedly green fluorescent molecules with a correctly chosen requires depending on how you do it, this can require slightly careful timing. So this is why it's expensive. So you have to have, in a simple way, you have to have a pulse of green laser light, and then you have to have a pulse of red laser light, which has been shaped into this red donut. And after that, you have to switch on your photon detector. And you have to do that at a reasonably fast pulse rate, like 80 megahertz or something. So it all costs money. But if you can do it, then the STED, or stimulated emission depletion principle, is this one. That here's your diffraction-limited spot of laser light. If you use this depletion beam to take away most of it, and this is not prevented by diffraction limitation, then what you're left with is effectively a much smaller spot of laser light, and then you collect the image data from that spot. This gives you basically a confocal microscope with a much smaller pixel size, and that's very useful. The way that you set this up optically is, as I say, you need it's moderate. It's fairly complicated. It's probably the most complicated optically of the super resolution techniques to set up, because storm you can do basically with nice high power illumination in a wide field or a turf microscope. STED requires also a confocal microscope itself is a bit more expensive than a wide field, but you need your confocal illumination optics for conventional illumination, and then you need this depletion beam, and you have to align the depletion beam to be perfectly centered and perfectly circularly symmetric, and you need to align it onto your sample, and then possibly you need some detector which has a time-gated operation, but you don't, because you can get around that in other ways. But regardless, in principle you need a high-power depletion laser, typically a TISAF laser, so it costs money. But if you have that, then you can take nice pictures. Here is an old one from Stefan Hell, who developed the technique. So this is a blurred confocal image of some sort of structure in a brain cell, and then here is the high-resolved STED reconstruction of that. Do I have a second STED? Yeah. And if you want to, as I say, it's important to make quantitative conclusions about your data. If you have, here we have a confocal microscope image of some fluorescent beads, and the corresponding STED image of the fluorescent beads. So the resolution is much better in the STED, and by taking a cross-section between two of the beads in this image, you can see that it's a single blur in the confocal microscopy, and the cross-section profile through the STED data shows that you have this double peak of intensity. So that says that even with a bead spacing of 40 nanometers, you still have the ability to resolve them apart. So then that means that with STED we can access the sort of single protein dynamics level of length scale. And we can do that using a light microscope, so we can do that then in living cells. Okay, so that's very useful, but it costs money. But nonetheless, we can do that. We can do that now. So here are some of these possibly antibiotic-resistant spores which I described earlier. And here is what we've done is we've labelled one of the coat proteins on the outside, which we're interested in. This one, coat Z. This is a sticky protein, I think. This causes. Graham's going to watch the video of this and he's going to tell me it's not. Coat Z is an outer crust protein. It causes the spore to stick to things, possibly. And so we want to know where it is. So we put make a snap fusion of coat Z, and then we make that fluorescent. We take a conventional confocal image of the spores, and they look like this. And I've just color mapped fluorescence intensity onto that instead of black and white because it's more interesting. And so that's conventional confocal, and we get a much better quality image by using STED to look at this coat. So we see much more clearly that we have just the outside of the spore is fluorescent. That's where the outer crust is. And this is very good. It means that we can no matter what the sample is, we can take a high resolution picture of it. But probably going to stop for a few questions and some coffee there, because after that I'll come back and talk about methods which I think are more useful mathematically, because this is a high resolution imaging, is a high resolution image, but so what? What I want to do is I want to make conclusions about which protein is where inside the bacteria cavity. And we haven't actually made that conclusion. We've just taken a very, very good quality picture. So in order to make more quantitative conclusions, I want to use a mathematical analysis on this type of data. I'd like to come back to that after we have a break for coffee, so that I can do all of the maths and inference at once. Okay? So if that's okay, then we'll pause on that slide for coffee and for any questions that you want to ask at this point, if that's okay.
You go back to some like 3 slides. And
More? Yep.
yes, and more.
Yep.
Yeah, this one.
Yeah.
You could explain how the light goes.
How the
Light. Like there are 3 waves.
I think I can. So what do you know already about epifluorescence and confocal fluorescence microscope setup? Would you like me to outline that, or do you want me to do something more complicated?
For example, here where is the incident rate?
Okay, sure. So I've color coded this. So the blue and it comes out as brown, but it's meant to be sort of red. The brown light and the blue light are both the illumination. So they're being focused. So they're being reflected off of suitable dichroic mirrors.
The green is reflected.
Sorry, yeah. So I've got you. So the illumination is the normal illumination is the blue. That corresponds to this diffraction limited green stuff there. And the brown light is the depletion beam. So that corresponds to what I have in sort of brownish red there. The colours have been slightly adjusted by my version of PowerPoint, as in they've been completely flipped. And then the green light, in this case, is actually the fluorescence which is being detected. So I'm not following the convention that I was using on the previous slide.
Yeah, so the incident rate is like incident light differences.
That's right, yes, that's right. So you have probably your illumination. So it's color-coded, so illumination I've color-coded blue. Actually the normal illumination is something like 650, it's basically a red light, and then the depletion beam is something like an infrared light, something like 780 nanometers if it's a TISAF laser, and then the fluorescence is somewhere in between them. But yeah, but ignore my color. Well, my color coding is varying on this slide. I'm going to blame PowerPoint for that. So normal illumination in blue, depletion in brown, and then fluorescence in green. Yeah.
And how is the alignment between the excitation and reflection being checked? Because it's very critical.
Oh yeah. So that's a good question. So one of the ways that you can do it is that you have a sample like these beads, so you disperse something like something that's. So one of the other drawbacks of this technique is that because it's very high power laser, it's extremely destructive. So it's a bit like looking at the sample with the eye of Mordor. So you get good results once, and then you've completely destroyed what you've looked at. So you need some sort of essentially indestructible calibration sample. So you have some gold beads, and what you can actually do is you can cheat and look in a reflected light mode. So you can focus it with the normal laser, and then with your what's supposed to be your depletion laser, which should be a perfectly circular aligned donut shape, you can switch to just that laser and check that you get in the right detection mode that you get the correct shaped circles. Alternatively, you can sort of if you have some beads and you know they're just points, you can iteratively adjust you can iteratively adjust the XY position and the shape of your depletion beam to make sure that your stead image of what you know is beads comes out correctly centered and as bright as possible. So the PhD student in the acknowledgements, I will point out, knows much more about this because he's the stead expert. But essentially, it is a process that you have to get your alignment right before you start, probably before you start for the day with this technique. Is that okay?
Yeah.
The expectation of the course, just in vibration state.
Electronic probably. The
Excitation of the course, just only in the vibration state, or can take.
I'm not a spectroscopist, but I think it's an electronic transition typically.
Or can be in the rotation state.
I'm pretty sure it's an electronic transition, but I think it's not. Ask me a coffee. I don't want to answer that one on the camera. Electrons are definitely involved, but it probably depends on the molecule and what sort of things you want. Okay. I think we might have exhausted my knowledge of STEM about 10 minutes ago.